Yes.
Okay. Can you see my pointer? I'm moving my pointer now. Can you see it?
Yes, we can see it. Yes.
All right, good. Hi. Yeah, so I'm Peter Lee. Like the previous two speakers, I'm also a physician scientist. I chair immuno-oncology at the City of Hope, which is a cancer center in the Los Angeles area. And I've been studying the immune system in cancer patients for over 25 years. I'm a big fan of mathematicians, and I'm also a strong believer that we need mathematical and computational approaches to supplement what we do on the bench to really gain mechanistic insight. So I'm very interested in feedback from the group and hopefully also working together. So actually, I think the last speaker used the term hot tumors. So what makes tumors hot? That's a very important hot question in the field of immuno-oncology. And certainly something that a lot of people care about because if you can make tumors hot, they tend to respond better to immune-based therapies. So the full talk would have had two components, a part about aspects of the tumor microenvironment that regulate whether the tumor is hot or not. And then there's another component which is very important in the tumor-genating lymph nodes, which is actually, I would argue, just as important, if not even more important. But given that we only have 15, at the most 20 minutes, I think we'll only be able to cover the first part, the tumor part. So what is a hot tumor? The hot term is basically a very simplistic term that refers to immune infiltration. So a tumor that has immune cells inside are basically considered hot, and a tumor that does not have immune cells considered cold. And the reason why it's important is because there's been a number of studies showing that prognosis is better when the patients have hot tumors and also responds to immune-based therapy, particularly immune checkpoint inhibitor therapies, usually associated with hot tumors. And right now in the field, the dominant view is that tumor mutation burden is the primary driver of whether tumors are hot or cold. And so that's the term suggests, is tumor mutation burden is simply a reflection of how many mutations are in that tumor. So have there been a number of studies trying to look for biomarkers for efficacy of immune checkpoint inhibitors, ICI is immune checkpoint inhibitors. And the two that have really come out to be significant are, in fact, tumor mutation burden and also immune infiltration. And the assumption is that these two, because they are the two most dominant biomarkers, they must be linked to each other. And that kind of makes sense, right? So because when there are more mutations in the DNA of cancer cells, when they're in the coding region, they can code for so-called neoantigens, novel antigens, that could be presented on MHC molecules to the immune system and drive an immune response. And in fact, the US FDA has approved the tumor mutation burden as a biomarker to predict response to immune checkpoint therapy, specifically for anti-PD-1 therapy. And the threshold there is greater than 10 mutations per megabyte. So there are certain tumors that have fairly low tumor mutation burden. And a pretty common example there is breast cancer, and particularly ER-positive breast cancer. And then there are other tumor types that have high TMB, such as melanoma. But in fact, few studies have focused on the direct relationship between TMB and tumor infiltrating lymphocytes. But if you look back, even back in the mid 2010s, 2016, 2017, there was the first paper kind of raising doubts about this relationship. When they looked at T cell tumors with either high T cell signature or low T cell signature, in fact, they had almost equivalent tumor mutation burdens. And in this paper, they actually found that rather than TMB, the most direct correlation to T cell infiltration is actually a type of dendritic cell that's BAT-F3. So this was really the first instance where TMB was not viewed to be associated directly with T cells and tumors. But despite this paper being around for about 8 years, it's really not made a dent in the field. So most people today still view TMB as the primary driver of T cell infiltration. So we want to take a deeper look at that, especially now that we know so much more about T cells in tumors. So here are 2 papers that came out within the last 5 years, asking the question, are T cells in tumors tumor-specific? And using some of the new techniques to identify tumor-specific T cells, the answer is actually surprisingly that the majority of T cells in tumors are in fact not tumor-specific. So that this paper called them bystander T cells. And in this paper, they estimated only about 10% of intratumoral CD8 T cells are in fact tumor-specific. So the other 90% are bystander, and they're specific for something else. And presumably, tumors create exert inflammatory signals which drive T cell infiltration, and they don't necessarily have to be tumor specific. They could be just activated T cells from a virus infection or something else, and that's presumably what's happening here. So additional papers have identified a marker CD39 that seems to identify tumor-specific T cells. So for those of you who may have heard of CD39, it's one of the ectoenzymes that cleaves ATP adenosine triphosphate into adenosine. So it's one of the phosphatases. It turns out that ATP in tumors can be very, very high due to high turnover and cell death of cancer cells. And ATP is a very inflammatory signal to the immune system, whereas adenosine is an immune suppressive signal. And so there's actually a lot of immune regulatory mechanisms to convert ATP into adenosine. So basically turning an inflammatory signal into an immunosuppressive signal. So CD39 is an enzyme that cleaves ATP and adenosine. There's another one called CD73. So CD39 is important. And another very important concept in the field of immuno-oncology is the so-called exhausted T cells. I'm sure you've heard that term, exhausted T cells, or TEX. And that refers to the notion that T cells have lost their normal function of producing lots of cytokines and killing, and have turned to either not doing anything or doing something else. And so they're called exhausted T cells. I actually do not agree with that term. We and others have found that exhausted T cells are in fact metabolically highly active. So they're not exhausted. They're not just sitting around doing nothing, but they've been epigenetically reprogrammed to do something else. And that something else is no longer producing cytokines or killing, but something that we don't fully understand yet. But one of the things that they seem to do is to make a chemokine CXCL13, which is a strong B cell attractant. So I think B cells will play a very important role in tumors that we don't fully understand yet. But exhausted T cells also express CD39. And so putting two and two together, I think we and others believe that there's a subset of T cells within tumors that are exhausted that express CD39, and those are the actual tumor-specific T cells. And through single cell sequencing, we've developed a specific gene expression signature for exhausted T cells, human tumors. And we're not the first ones. There have been other groups that have also done single cell sequencing and have developed exhaustion signatures from other human tumor types. And so we validated our signature against other signatures from melanoma, lung cancer, and found that they all actually correlate very nicely. So it seems like exhausted T cells within human tumors share very common biology. So they have very similar gene expression patterns. So using that, we embarked on a computational project led by two people, Hui-Hua and Joyce in my group, who are primarily computational experts. We took advantage of a large public data set from TCGA. I'm sure you've heard of it. And that's a huge public data set. And we primarily focus on 9 major tumor types as listed here. And we took advantage of both RNA sequencing data to look at gene expression profiles, as well as whole exome sequencing data, from which we can then determine tumor mutation burden. And then we did various different computational analysis, including gene deconvolution analysis. And all this work just came out earlier this year. So if you're interested, please take a look at our paper in eBiomedicine. So we wanted to ask, is there any relationship between exhausted T cells, which we would argue is really the most specific cell type that are tumor specific, against TMB? And so looking at our exhaustion signature against TMB, we found essentially no correlation in any of the tumor types. So basically a flat line across. And so here's just a higher example of all of the different tumor types. We also went further to look at neoantigens. So there's, as I mentioned, so DNA mutations in the coding regions could lead to neoantigens. And so there's a large neoantigen database available. So we looked at that database and we did correlation between neoantigens against exhausted T cells, and again found a very weak or almost no correlation with T cells, Ts in tumors. So we then went back to these data sets and we put together 10 what we consider key immune parameters in cancer, including TMB, exhaustion, but also a number of different immune markers such as dendritic cells, different types of T cells, interferon, but also something called STING, which I'll get to in a minute. And by using hierarchical clustering, we found that there were 6 clusters. And there's 3 patterns: a low TMB pattern, which have clusters 0, 1, and 4, intermediate and high TMB clusters. And so I wanted to especially focus on cluster 4, which is a low TMB cluster, but yet has moderate to high T cell infiltration. And what we found there was that there's a strong relationship between the exhaustion signature and STING signaling, but not with TMB. So what is STING? So STING is actually basically like a fire alarm inside a cancer cell. It turns out that is extraordinarily well preserved through biology. In fact, there's even evidence that bacteria have this STING pathway. And basically in human cells, the STING pathway detects cytosolic DNA. And when there is cytosolic DNA, the STING pathway gets activated. And that leads to production of certain antiviral molecules, because cytosolic DNA is generally a feature of virus infection, but also in cancer. And that leads to production in interferons, especially interferon beta. And so this is a way that a cell could alert the immune system that something bad has happened to that cell. It has cytosolic DNA, which could be reflective of either virus infection or cancer, and it sends out an alarm through interferon to alert the immune system. When we looked at TMB high versus low tumors, TMB high tumors, as you would expect, has a number of inflammatory immune markers, and TMB low tumors have not only STING, but are high in DNA repair mechanisms. So that really, again, clued us in that there's a relationship between STING signaling and DNA repair mechanisms. And so that's reflected here. There's, as you know, in cancer research, there's really a lot of great work done on DNA repair mechanisms in cancer. And so when we looked at the relationship between DNA repair and STING, what we found was that the higher the STING, the lower the DNA repair mechanisms. And here are just several examples of different, here's nucleotide excision repair. Here's another mechanism, replication stress response defect. And one other one, or another important relationship is apoptosis. So we also found a strong relationship between apoptosis and STING. So the picture that comes together is that when cells are defective in DNA repair, they're dying, that leads to cytosolic DNA, leads to STING signaling, and that leads to inflammatory signals to the immune system, which drives T cells into tumors. And lastly, we did a bunch of computational and statistical analysis. Here is a leave one out cross-validation showing that STING was by far the strongest predictor of T cell infiltration into tumors, much more so than TMB and even other immune characteristics. So because all of that work was done using the TCGA data set, we wanted to validate our results using another data set. So another data set that's widely available is the Metabreak data set, which comes out of the UK. And this nicely validated that there's a very nice correlation between STING signaling and exhaustion within tumors. We also wanted to look at metastatic samples because most of the tumors that we've looked at so far, both in TCGA and Metabreak, have been the primary tumor. But what about metastasis? And here is a public data set specifically for metastatic melanoma. And we also found a very good correlation between STING and T cells exhaustion. So it seems like this pattern is also consistent between primary tumors and metastasis. And then lastly, we did, because all of the analysis have been using public data, we experimentally validated using our own patient samples at City of Hope. In this case, we used 31 samples from human breast cancer patients. These are ER-positive breast cancer, which are generally of low TMB. And we did genomic sequencing using a platform called NanoString. And we did continue to find a very strong correlation between STING and exhaustion. We've went further to do single cell sequencing, in this case, 9 additional samples. And we focus on the cancer cells and have found that cancer cells with high TDX tumors have much higher STING signaling. So just to conclude then, that cancer genomics drives tumor mutation, tumor infiltration by immune cells, but not TMB alone. That even in low TMB tumors, there could still be T cell infiltration. And by the way, they could still respond to immune checkpoint therapy. And in those instances, STING signaling can compensate for low TMB and drive T cell infiltration. And STING signaling seems to result from high apoptosis, releasing cytosolic DNA, and also defective DNA repair mechanisms. And so STING looks like it's going to be a very significant biomarker for immune infiltration and ICI therapy response. And that also points to the possibility of modulating STING signaling to further enhance immune checkpoint therapy. I would add that there are already STING agonists out there. Many of them have been in clinical trials with actually modest at best clinical responses. But these have not been very effective agonists. So in my lab, we're really trying to better understand how DNA defect mechanism, repair mechanisms lead to STING activation. And we're hopeful that we can come up with better strategies to activate STING signaling specifically within cancer cells. So I just want to end with this slide to remind us that the tumor immunity cycle is complex. So we focus on the tumor microenvironment, and most other labs do as well. But in fact, a lot of the immune responses actually happen in the tumor-draining lymph nodes. And we have studied the tumor-draining lymph nodes for 15 years, and there are many immune changes that happen in these tumor-draining lymph nodes that are important. So there's another part of the story. In fact, that work just got accepted in Nature Communication just last week, and so that'll be coming out. But because of time, I won't be able to talk about that today. So I'd like to end here, and I'd like to take any questions or comments. Thank you.
All right, thank you, Peter.